So good evening, everybody, and welcome to the Royal College of Surgeons for me at least, it feels like the first wintry night. It's really dark and miserable and we all want to be home in our PJs, but anyway, we're here. So you're very welcome to what's, I guess, become our annual public patient interaction evening to talk about brain cancer research. So you're all very welcome and welcome back to those of you who were with us last year. My name is Professor Annette Byrne and I am head of the Precision Cancer Medicine group here in RCSI. I've been at RCSI for 15 years and we first established the group pretty much agnostic about what cancer we were working on. So I was a tumor biologist. I'd come back from the States. I'd worked on various different cancers. And as time went by, little by little, I got interested in two cancers in particular. The first is colorectal cancer, and the second is the cancer we're going to talk about this evening. It's brain cancer. Every year when I introduce this, I make the point of saying that I have a personal interest in these cancers. Sadly, my dad passed away from colorectal cancer many years ago, and my mom passed away sadly from glioblastoma. So I think it was naturally in the back of my mind that these were the two cancers that I was going to be driven to work on. So this evening, I'm going to introduce you first to my group, many of whom are here. And whoever's in my group that's here, you might stick up your hand and just wave, because afterwards we'll be around to answer any questions you guys might have on any of the research we're doing or any of the programs. So with that, I will, so we are the Precision Cancer Medicine group. We're based here in the RCSI, and today we're going to focus on our brain cancer work. This is me. So as mentioned, we have two subgroups. The first there on the top are the people that you're most interested in talking to this evening. And this is the brain cancer subgroup. And Dr. Kieran White, Kieran, give away, is here tonight. He's going to be speaking about a major paper that we had published last year in glioblastoma. And my PhD candidate student, Mohammed, I don't know if Mo is here as well. Mo has just joined us from Jordan. Very, very happy to have Mo on board working on a brain cancer project. So Dr. Suzanne Murphy probably, Suzanne's here, I didn't know she's in the hospital. Suzanne is a neurosurgeon who's doing her MD with us. Dr. Kate Connor, formerly subgroup lead of the brain tumor, but she absconded to Trinity. We'll get her back. And Mr. Kieran Sweeney, who's a neurosurgeon. Kieran's down the back. Kieran's our lead clinical lead. Kieran is a consultant neurosurgeon at Beaumont and also will be speaking. Also, and not to forget about our people who work on colorectal cancer, Dr. Ian Miller, who give you away there. Ian also was a bit of a jack of all trades. I think, Ian, at one point you worked in GBM as well as the States. Yeah. And Colin Connolly, the research assistant, and Mr. Liam Shields, who is here. Liam is kind of in the middle of it all. Liam is a very important person who works both on brain cancer and on colorectal cancer. And we're very grateful to Brain Tumour Ireland for part funding Liam's work as well. And then probably the most important person even beyond me is Dr. Alice O'Farrell. Alice is there at the back. So Alice is our research program manager. So Alice brings it all together. So as mentioned, we're going to focus tonight on our brain cancer work. And before I sort of introduce the talks, I wanted to say a little bit about what our group does. So we are essentially a preclinical research group. That means that we work with models of brain cancer, both in Petri dishes and also animal models. Our focus really is trying to understand the underlying biology with a particular interest in glioblastoma in grade 4 brain tumors. We want to understand how the disease works. We want to understand why the disease has this ability to become resistant to therapies. So it seems that for decades, us in the lab and also clinicians have really been trying to treat tumors in many different ways. And the tumor is very clever. It always seems to have this ability to become resistant. So we're trying to figure out different ways of overcoming this resistance. Let's call a spade a spade. Our ultimate goal is to identify new treatments. So whatever we do, we have the patient in our minds. And that is why we work very closely with our clinical collaborators, Kieran and Suzanne, to really guide us in making sure what we're doing is clinically relevant. We don't want to be one of those research groups who's stuck in the lab and in a fussy little lab and not thinking about the patient. So we really enjoy events like tonight where we get to meet patients and their families, and we really engage very closely and a lot of the time with our clinical partners to keep us on the right track. So some of the cool stuff that we get to play with, I guess, are our group has particular expertise in imaging and multiomics. And just to say a little bit about imaging, imaging is kind of a neat thing to talk to members of the public about, because I think we all have an understanding of what medical imaging is. So I guess most of us have either had a CT scan, an ultrasound, or an MRI at one point in our lives. So what we have, but engineers in the field have been able to do is to develop imaging modalities or imaging scanners that are basically miniaturized, so we can use them with animal models. Why is this important? This is important because what we are trying to do when we model cancer is try to the best of our ability to replicate what a patient sees. So the treatments that the animal models receive, the imaging that they may undergo, we're always trying to replicate what the patient sees. This is important because we're not trying to cure a mouse tumor. We're trying to cure a patient tumor. And based around the limitation that a mouse is not a human, we're trying to the best of our ability to mimic the patient in our models. So what you see there is an optical imaging scanner. So that picture there, the colored one, is actually a mouse with a brain tumor. And in this case, we've manipulated the tumor such that it can be picked up with light. And this is called bioluminescence. So we have a scanner that can detect light. And you can imagine as the tumor grows or shrinks, if we have a good treatment, the light signal will go down. We also see there an MRI scanner. So we have a new scanner that's we're going to kick off our first scans on a mouse MRI scanner. So many of you will have had backs or, looked at in an MRI scanner. It's a horrible experience going into that tunnel. So imagine what that is miniaturized. So we have one of those scanners that is miniaturized, and you put the mouse in on a little bed and they go in for their MRI. So again, we're able to replicate in the animal model, we're able to replicate what the patients see. Kieran is going to talk a little bit about multiomics. It's kind of a bit of a complicated thing. But really what we're trying to do is look at the genetics surrounding brain tumors, trying to treat different subtypes or different categories of brain tumors, depending on the genetic profile or the protein expression patterns of tumors. And so we have to use different technologies to do so. Ultimately, we are trying to develop sufficient data in the preclinic. So in the lab, if you like, we're trying to develop enough data that will support future phase 2 clinical trials. And our goal would be in the next 2 to 3 years, we would hope to be able to initiate a phase 2 trial with our clinical collaborators out at Beaumont. So it's a very busy time for us. We've had, since our last meeting here almost a year ago to the day now, we've had some significant success in terms of getting our work published, which is international recognition that we're on the right track. I'm also very grateful, and I want to point out that we've been very successful in gaining funding for our research. As you can imagine, this type of research is extremely expensive. So we've received money from the EU, from the Health Research Board, from SFI, from Brain Tumour Ireland, and from Beaumont. So all of this money goes into a giant pot that Alice makes sure I spend correctly. And we set out on a mission to develop sufficient supporting information and supporting data to allow us to move into a clinical trial with our work. Yeah, that's basically the what I want to say in introduction. So I'm delighted we have a varied agenda for this evening. First up, I'm going to invite Dr. Kieran Sweeney, who's going to tell us specifically about his work and about the big paper we had published last year. Kate Connor is then going to tell us about the new work that she's doing at Trinity, looking at a very interesting project looking at ways to treat brain tumor related epilepsy, which is something I knew quite little about until Kate got into this work. I'm delighted to also introduce Professor Claire Gardner from Trinity College, who's going to talk about next generation immunotherapy in GBM. And then our clinical collaborator, Mr. Kieran Sweeney, is going to talk about drug delivery methods to brain tumors. So with that, I will stop. At any point throughout, please just put up your hand if you have any questions whatsoever. And then I think they're feeding us afterwards, are they? Yeah, good. So we'll be around if anybody has any questions. So thanks very much, everybody. I hope you enjoy the evening.
Yeah, brilliant. Yeah, so I carried on in Net's lab to sort of continue the work that we've been doing for the last few years. And today I'm delighted to present some of the work we've been focusing on throughout the PhD and ongoing work, where we're really trying to identify novel precision treatments for glioblastoma. So for this presentation, I'll give an overview of glioblastoma and its biology and really the challenges we're trying to overcome for more effective treatments for glioblastoma. And then I'll be focusing on one of the projects where we're identifying novel treatment strategies for GBM. And then I'll discuss our ongoing projects and future directions within the Precision Cancer Medicine Group. So it's important to firstly know how cancer is formed. So in normal cell development, you have normal cell which divides by a process called cell division. And this is a really structured and organized process which forms healthy tissue. However, the differences with cancer or this abnormal cell growth is that there's these distinct differences from the normal cell when they divide, creating a malignant or cancer cell. And these all have distinct properties called hallmarks of cancer. And this allows the cancer cell to have this uncontrolled growth and uncontrolled division, forming malignant tumors such as glioblastoma. So therefore, it's really important to understand these hallmarks of cancer for more effective cancer treatment. So specifically, glioblastoma is the disease which I was focusing on throughout the PhD. And this is the most common and aggressive form of glioma in adults. And its typical symptoms are headaches, cognitive changes, seizures, and focal neurological deficits and weaknesses. And it's slightly more prevalent in males than females, about 55% to 45%. And its peak incidence is 50 to 65 years. And there's approximately 300 people diagnosed yearly with glioblastoma. And the current standard of care for glioblastoma is called the STUT protocol, and this was established in 2005. And this is where patients have surgical resection of the GBM tumor, and then followed by chemotherapy and radiation therapy, and then followed by chemotherapy as a single treatment. However, there needs to be more treatment options and more specific treatment options for glioblastoma patients because a lot of GBM patients don't respond to this care or become resistant to the standard of care therapies. And some of the challenges which we really need to try and overcome, specifically with such an aggressive tumor such as glioblastoma, is this highly invasive, which means it can infiltrate into healthy brain tissue. Also, the blood-brain barrier poses as this physical barrier, which makes it very hard to target the brain with treatment. It's not only resistant to standard of care, but is also resistant to novel treatment strategies as well. And then the two sort of areas which I've really focused on over the last few years, and we have with the group, is a heterogeneous GBM, which means that each GBM tumor has distinct differences, making it difficult to treat, and also this tumor microenvironment. So the immune system is a really important defensive mechanism for the body, and it sort of acts by, it detects foreign material or pathogens in the body, and this initiates an immune response. So it signals to the immune cells that we need to target this potentially harmful material, where it'll go and attack and destroy this material or these harmful cells. However, what can happen with these solid tumors, and in particular glioblastoma, is that GBM can actually create an environment which allows the tumor to hide or evade from the immune system. So it can sort of hide from the immune response. So as you can see in the diagram here, you'd expect these immune cells to sort of attack the glioblastoma cells. However, what it can do is the tumor can actually make these immune cells become exhausted, so they can't actually attack the tumor cells. But they can also change the properties of these immune cells. So they can actually cause these immune cells to promote tumor growth. And what also they do in this tumor microenvironment is they can actually form blood vessels, and it's a process called angiogenesis, which supplies the tumor with oxygen and nutrients for this continuous growth of glioblastoma. Also, another challenge which we're trying to overcome is GBM heterogeneity. And this really means that even though it's identified as glioblastoma as one tumor, you'd think they'd have similar properties, but you can see different tumors in different GBM patients have distinct properties, not only genetic alterations, so mutations or changes to their DNA, but different tumor microenvironment profiles. So there's distinct differences in such as the number of immune cells, the function of these immune cells and blood vessels. And also these different GBM tumors can respond differently to treatment. So therefore, it's really important for us to try and understand and try and identify more personalized, effective treatment strategies for each GBM patient. And also these challenges have resulted in limited response to new treatment strategies. For example, immunotherapy, which has shown a lot of success in other solid tumors. So we really need to understand why there hasn't shown the same success in glioblastoma. So immunotherapy is a type of cancer treatment that helps your immune system fight the cancer. And it can do this by boosting how the immune system works so it can find and detect and attack these cancer cells. So one example of immunotherapy is immune checkpoint inhibitor. And just in this diagram, you can see the tumor can actually bind to immune cells to deactivate them or turn off these immune cells. However, if you introduce these immunotherapeutic drugs, they can block or almost release the brakes on these immune cells, causing the immune cells to turn on and activate and attack and destroy these harmful tumor cells. And there's a lot of different immunotherapy strategies in cancers, which is really targets manipulating different parts of the immune system. And these are all being currently investigated in several cancers, including glioblastoma. However, for such a heterogeneous disease such as GBM, it's really important to try and identify the patients which may most likely respond to immunotherapy in this disease. So this was really the premise and sort of where our work was based on. And we recently published this work in Annals of Oncology. And the main aim was to identify new GBM patient subgroups to help direct treatment approaches for GBM. So to do this, we received tissue from a cohort of glioblastoma patients. And what we did then was we wanted to perform molecular analysis. So we really looked deeper at the glioblastoma tumors across the cohort to really understand the mechanism of these tumors and to see what was causing this aggressive nature of GBM. And what we found from this analysis is that the glioblastoma could be subtyped into 3 distinct groups based on their tumor microenvironment. So what we saw was a subgroup which we identified as TME low, TME medium, and TME high. So the first thing we did, we wanted to really understand the biology of these TME subtypes. And what we could see was the TME low subgroup had a very low number of immune cells. So we call this an immune cold subgroup. But what it had was an enrichment, an increased number of these invasive and these aggressive glioblastoma tumor cells. TME medium was more of a heterogeneous subgroups. So we saw immune cell populations, but we also saw an enrichment of this blood supply to the tumor, an increased number of these neurons, so these signaling, important signaling cells in the brain. And then TME high had a very large number of immune cells, but it was both good immune cells, which can attack the tumor, but also these immunosuppressive cells, which almost have the brakes on. They also had different mutations present within these TME subtypes. And just for an example, we noticed and identified that TME low had this specific alteration to the DNA, which caused this low number of immune cells present in this subgroup. And then we also looked at the treatment-resistant mechanisms. So we looked at these tumors based on their TME subtypes before they received current standard of care, and then after they received the current standard of care treatment. And this is really important to understand, to see what mechanisms the tumor is actually having to evade this treatment pressure. And that'll give us an idea of how can we actually target this tumor to have an effective response. And then importantly, we looked to see at whether these subtypes responded differently to immunotherapy. So we assessed 3 immunotherapy clinical trial cohorts. So the patients received immunotherapy, and then we applied the TME subtypes. So TME low, TME medium, TME high. And what was really important from this work is that we we observed that the TME high subgroup had an increased response to immunotherapy treatment and also an increased number of these immune cells which can actually attack the tumor. And this is important because there's been a lot of sort of failed immunotherapy clinical trials where we see limited response. But this really emphasizes the fact that immunotherapy does show potential in GBM, but we really have to be specific on the patients where it can respond to. So from all this analysis and from all this sort of multi-omic work, we sort of proposed treatment approaches for the 3 subtypes. So we hypothesized for TME low that they would benefit from chemotherapy and FAK inhibitors or tyrosine kinase inhibitors, where you're really trying to target these aggressive glioblastoma stem cells. For TME medium, we really want to, we believe that using immunotherapy and anti-angiogenic agents will be beneficial. So this will really try to attack, to sort of promote the immune activation, but it will also try and suppress this these blood vessels supplying the tumor for the nutrients and oxygen to grow. And then finally, TME high, we believe that if we treat with immunotherapy before and after surgery, so really trying to release these brakes for an immune response before and after surgery, will have benefit in this subgroup of patients. So building upon this now, as Annette mentioned, there's several sort of ongoing projects with the aim to further understand the biology of these TME subtypes and identify specific targets across the TME low, TME medium, and TME high subgroups. And the really purpose of this is to identify novel treatment strategies and also test our treatment hypothesis in the subtypes. And a real main focus then is to drive these, not only just to sort of hypothesize, but drive these into the clinic where hopefully we can see patient response in a phase 2 clinical trial. So I just want to say a huge thank you to, it was a huge collaborative effort, all this work. So a huge thank you to Professor Annette Byrne, who was driving this project, as well as the Precision Cancer Medicine Group and all the funding organizations. And most importantly, a sincere thanks to all patients and their families, for we couldn't do any of this research without them. And I just know as a group, we're really committed and really passionate to working on glioblastoma with really the aim to improve patients' lives. So thank you very much for listening. And if you have any questions now or after, I'm more than happy to answer. Thanks. Yeah.
Not looked that good in terms of clinical trials and immunotherapy. Do you know of any positive news in that respect, or what's? I know Kieran is off to Vancouver next week to the site of neuro-oncology, so you have all the fresh news. But
yeah,
you know,
so there's a few. Yeah, so there was sort of 3 main immunotherapy trials, and there didn't seem to be much benefit. But now, sort of more recently, there's appears to have benefit. So neoadjuvant. So what I said, having these immunotherapies before you have surgery, it really seems to increase the immune response for the GBM patients. And they showed, that was the sort of first trial where they showed immunotherapy response in glioblastoma. And also this other immunotherapy, which is oncolytic virus. This seems to be the most promising immunotherapy at the moment, where there's been 2 big studies now where they seem to have response like ours in a sub-cohort, in a subgroup of patients. So they actually applied these subtypes and they showed in one of the subtypes there was an increased response. So I think there is still definitely, we can still use immunotherapy for glioblastoma. We just got to be really specific and identify the patients that can respond. That's a good question. Yeah, so apart from I was part of the Glio Train program, and a student actually in RCSI, she was looking at the difference in the long responders versus the short, and I think she was looking at, I'm not sure if she looked at the immune profile in particular, but she saw distinct differences which they're currently investigating. In terms of our work, we saw an improved survival in TME high. So we can see that these sort of are predictive of response to immunotherapy. So it's something we need to look further into. And it's one of those things, the more sort of samples we can get and we can look at this data, the more we can know as well. Yeah. Yeah, so I think Kate would probably be better to answer this to me, but for basic, I think, but basically it's this protective barrier from the brain. So originally the brain was sort of really considered immune privileged, though there weren't many immune cells because they thought the blood-brain barrier. But what this does is really protective mechanism for the brain. So, but what it does, it means you can't, it's hard to deliver drugs through to the brain. So it has to be very small molecules so it can pass this physical barrier to the brain. So that's something we're trying to do. I know, Kate, you were looking at nanoparticles as one potential sort of therapy. And yeah.
Say more about the blood-brain. Barrier and can't pass the barrier, so it's very difficult to find treatment that you can give, a tablet or through an IV that can access the brain easily. Yeah, which is one of the difficulties that the you have to administer it. You can't administer it directly. There are ways to do that. There's a lot of pros and cons. It's there for good reasons. They're to protect the brain for infection and all the rest of it. But when it comes to then developing drugs for diseases that are in the brain, there's a problem getting over the blood-brain barrier. So it's a double-edged sword. Anyway, well done.
Thanks a lot.
Hi, everybody. So, so my name's Kate, and I'm currently a research fellow in Trinity College in the discipline of physiology. But I spent the last almost 8 years working in an ETS group, and my work's been on glioblastoma for all of that time. And now I've moved into the Cunningham Laboratory, which is run by Professor Mark Cunningham. And basically the focus of this lab is we study epilepsy, but we would also study how different therapeutic interventions or different drugs can affect brain activity. And what my project is focused on the development of new treatments for brain tumor-related epilepsy. So, as you can see, I worked in an S group for a long time. We have the same slide. That's my slide. I made that slide. So I'm going to give you guys an overview of brain tumor-related epilepsy. I'm going to talk a little bit about neurotransmitters in the brain because these are essential to normal functioning of the brain. But sometimes in the case of when you have a tumor, they can basically subvert or use neurotransmitters to fuel their own growth. It's also really critical to the development of epilepsy in the brain tumor setting. And then I'm going to talk about two different projects. So the first being my ongoing project looking at a novel way to treat brain tumor-related epilepsy, but also another smaller project which is ongoing, which is essentially a quality of life and economic cost study of brain tumor-related epilepsy. So BTURE is a really serious pathology because it comprises two very difficult to treat pathologies. So you have both oncology, or you have both a tumor and you have epilepsy. Seizures can be the first presenting symptom for many patients, but they can also manifest as a complication over the course of the disease evolution. There's a huge range of sort of frequencies of epilepsy developing depending on your type of tumor. And a lot of people can develop them following treatment as well as at the initial diagnosis. And it's very difficult to treat them. So a lot of the anti-epileptic drugs have what we call a very narrow therapeutic window. So the therapeutics, which means the amount of drug you need to give to affect the cells that are causing the epilepsy, sometimes what happens is you affect the normal brain cells around that as well, causing a toxicity. And there's quite severe side effects to many of the anti-epileptic drugs. In addition, these are given in combination with patients undergoing standard of care, as Kieran described. So they'll have surgical resection, they'll have chemotherapy, radiotherapy, and overall, there's a lot of side effects to this. So without going into too much detail, I'm not specifically going to talk about glioblastoma because depending on the type of tumor that you have, the likelihood of developing epilepsy associated with this tumor can be very different. So there are some types of tumors called DNETs, which 100% of patients will unfortunately develop epilepsy. If you have what's called a low-grade glioma, it varies between 50 and 90% of patients who can develop epilepsy. If you have a meningioma, about 45%, and GBMs is a bit lower, between 25 and 40%, and then brain mets is again about 20 to 40%. There's a lot of different factors which will influence the development of epilepsy, so it's not only the location of the tumor, but also the kind of molecules and proteins that the tumors are producing. And because each of these are different kinds of tumors, they have different reasons for causing this epilepsy. So the epileptogenesis, or the development of epilepsy because of a brain tumor, can be influenced by a number of factors, and two of which I'm going to just talk about to begin with is tumor location, so where exactly the tumor is, and the type of tumor that you have and whether or not it has an IDH mutation. So depending on where your tumor is located, you may have different types of seizures. So in green, if you have a lesion in the more frontal area, which is actually the part of the brain responsible for language control, or Broca's area, you have an increased risk of simple partial seizures, which are seizures where you don't lose consciousness. In the pink area, which is called the temporal insular region, which is basically a region of your brain involved in controlling like thirst and fatigue, you are more likely to have a complex partial seizure, so where you lose consciousness during that seizure. And then finally, in the blue area, lesions that involve what's called the premotor area, which is a part of the brain that's involved in movement and coordination, you're more likely to have a seizure that involves both of your hemispheres. So there's a lot of, as Kieran used the word, heterogeneity, so differences in the types of seizures you can have. So treating these seizures, as I said, is very difficult. So radiotherapy or chemotherapy can help reduce seizure frequency, but a lot of patients, oh, sorry, I made this on a Mac, so there's going to be some weird little bits going on here. So depending on while radiotherapy and chemotherapy can help reduce your seizure frequency, a lot of patients do continue to develop seizures or experience seizures. And actually quite a big proportion of patients with low-grade gliomas will continue to experience them after surgery, but also a large proportion of glioblastoma patients will develop them post-surgery. So it's a very complex condition that you have on top of the fact you have a tumor. And as I said, the current anti-epileptic drugs do have quite a lot of limitations. So for example, there's a drug called tolophenol, which just has had really mixed results. It works for some people, it doesn't work for other people. Then you have perampanel, which is quite effective, but has really severe adverse side effects with a lot of neuropsychiatric side effects. So it's really not a very great drug to be given. And then sulfalzine, which has been great in preclinical studies, but hasn't really translated very well to being effective in patients. But then when we talk about brain tumor-related epilepsy, it's really important to remember that the actual seizure as a symptom is just the tip of the iceberg. And patients who have brain tumor-related epilepsy, we have to remember there's huge effects which are not just related to it's difficult to treat and it's a symptom. So for example, it has huge effects so the adverse drug effects are a huge factor, but you have reduced cognitive performance, reduced physical and mental health, fatigue. Once you have a seizure, you can't drive, so you have a loss of independence, you become isolated, depression. And this is just a snapshot of the issues associated with this condition, and why it's really important that we are able to treat this condition. So basically, the fact that surgical, oncological and pharmacological treatments, so our chemo, our radio and our surgical intervention is quite limited in its efficacy for treating this condition, we really need to develop novel therapies that are targeted and that are better at treating this condition. And gene therapy is one of these approaches, which I'm going to talk about in a couple of slides. So before I talk about using gene therapy, I want to talk about what it is we're going to target using gene therapy. And so neurotransmitters are molecules or chemicals in the brain, which are perfectly normal and are incredibly important for normal functioning of your brain cells or your neurons. And these are essentially how your neurons will communicate with one another. Oh, sorry. So basically, when your neurotransmitter production can change because of the tumor, this can cause the development of brain tumor-related epilepsy. So when you want your brain cells to communicate, it's perfectly normal that neurons will communicate with each other through both electrical signals and then neurotransmitter production. And these cells are important to the normal functioning of the brain, and they work as a community. And basically, most people actually know about more about neurotransmitters than they realize that they do. So for example, adrenaline is a neurotransmitter. When this is produced and it's passed from one neuron to the other, you have what's called your fight or flight response. A lot of people have heard of serotonin, which is sort of your mood neurotransmitter, which is essentially a chemical that contributes to your happiness and well-being. And even endorphins are neurotransmitters. And the neurotransmitter that we are focused on and that we're interested in, that we know plays a role in the development of brain tumor-related epilepsy, is glutamate. So the normal role of glutamate is involved in learning and memory. and it is the most common, what we call excitatory neurotransmitter, which means it has a sort of a positive effect on the cells. It causes a signal to be passed through cells. And we know that glutamate plays a key role in brain tumor-related epilepsy. So normally, when you have two neurons communicating, you get an electrical signal that passes through your neuron, and then it will communicate with the next neuron by producing a neurotransmitter, and that next neuron will carry that signal onwards. But tumor cells are really smart. And what tumor cells are able to do is essentially communicate with a neuron in the same way a neuron is used to being communicated with by another neuron. And these tumor cells can produce neurotransmitters like you would expect a neuron to, and communicate with neurons. And we know that glioma cells produce really high levels of glutamate. So what happens is these tumor cells produce really high levels of glutamate in an uncontrolled manner. So neurotransmitters are like basically like when you want to plug something in, you don't want to leave it plugged in all the time. You want to turn it off so you don't burn out your light bulb, right? So neurotransmitter production and release has to be really controlled, but glioma cells are really smart, and what they do is they do not reuptake, or they do not break down their neurotransmitters they're producing. So they're constantly secreting this neurotransmitter into their environment and constantly signaling through the local or the nearby neurons. And what this results in is you get this overproduction and no absorption of these molecules, and you get uncontrollable excitability. So you basically switch on all the cells, all the neurons near those tumor cells, and that results in a seizure. And that uncontrollable excitability also results in the neurons dying after a while, sort of like a fuse blowing in a plug, and also allows the cells to invade more. So it's a really smart mechanism. They produce a molecule that's totally normal to the body in excess, don't break it down and recycle its components, and then turn on all the neurons, causing a seizure. So what do we want to do about this? How are we going to maybe interfere with this mechanism? So we are currently running a collaborative project between the lab in Trinity and Annette's lab here, and we want to use a gene therapy approach to treat this mechanism of brain tumor-related epilepsy. So we want to test whether a novel gene therapy can stop seizures using an animal model of brain tumor-related epilepsy, and then we also want to test if this treatment can be used in human tissue to prevent seizure activity in human tissue. So gene therapy is a method whereby you can engineer a cell to produce or stop producing a molecule of interest. Our molecule is called e-GluCl. I'm not going to keep saying that. I just have an E. So basically what we want to do is we want to engineer cells to produce this e-GluCl. So we have viral particles, so it's a virus. And what this does is it can insert a gene into your neurons. And this gene gets produced, you get your E's produced. And what this does is it basically stops the tumor cell glutamate from affecting the neuron. So it's a bit like having a fuse box. When that protein is produced in the neuron, it senses this really high level of glutamate, and the fuse box goes, you trip your switch, and it stops. So you don't get that excitability, and you don't get the propagation of that signal, and you don't get a seizure. So what we're doing is we have a mouse model, and these animals have tumors, and they have spontaneous seizures like you might see in the clinical setting. And we have a method where we can monitor their brain activity using telemetry. So like you would in a patient, you can put little probes on the skull, and you can measure the brain waves, and you can detect a seizure happening or not happening. So what we want to do is we have these animals who have tumors, and they have this little device to monitor their brain activity. We treat them with the lentivirus particle, and then we essentially monitor if they're having seizures or not pre and post therapy. So we can judge how effective the therapy is at dampening the seizure activity in the animals. We're also monitoring to see if that treatment can slow down tumor growth, because there's less excitability in the neurons, less neuron death, and less cell invasion. So ideally, the treatment should dampen the seizure activity, but also reduce the tumor growth.
What's in the guy that you guys want?
What's it? I don't know. I think he has, I think it's a lollipop.
Okay.
I wasn't sure. Or a whistle, or maybe it's spirometry. I don't know. Yeah. I couldn't find a better picture. I didn't have time to Photoshop it out. No. So the second part of this project is to see if this gene therapy approach can work in human tissue. So we are going to collect some what we call peritumoral tissue. So the tissue on the edge of the tumor, where the tumor is interacting with the neurons and where we think it's producing this neurotransmitter glutamate. And we're going to infect or engineer the cells using the lentivirus, using our gene therapy. And once these cells produce our little protein, we're going to measure the electrical activity or the seizure activity of these brain slices, a little tiny piece of brain. And we have this big fancy setup, which is I'm not going to go through, but it's basically a really cool setup where we can insert an electrode and measure the electrical activity of our cells, a bit like a telemetry EEG system we use in the animals. And what this will allow us to record is whether when we treat, does this tissue have a seizure, or do the seizures get dampened after the treatment? So the second project I'm just going to give a little bit of info about is a quality of life and economic cost study that's been ongoing. So this comes back to this sort of iceberg idea that, again, it's not just about the tumor, it's not just about the neurons, it's about the entire effect this has on people's lives when they have seizures. So brain tumor-related epilepsy is a major risk factor for long-term disability because of both negative impact of seizures on quality of life, but also as I said, the quite adverse side effects of a lot of the anti-epileptic drugs. And despite this really substantial impact to patients, the economic cost of BTRE and the impact on quality of life has never really been directly quantified in Irish patients. So we have a project, an interdisciplinary project, which is ongoing. Sorry for the poor formatting, my Mac. But basically, we have a team of people. So we have Professor Mark Cunningham, who's an electrophysiologist. There's me, who's kind of a preclinical oncology person. We have Kieran Sweeney, who's a neurosurgeon. We have Dominic Trappell and Rachel Kane, who are health economists. Rachel Kane is a really great undergraduate student who did a project on this over the summer. So the idea behind this project was to develop and estimate the impact of brain tumor-related epilepsy of economic costs in Ireland. So, and in the long term, the idea would be that you can judge how costly this is, both to the economy, but also the quality of life of patients, and help inform the allocation of healthcare resources. So Rachel developed a model, which is called a Markov model. And basically what this is a model they use in economic cost prediction, which is a bit like Google predictive text, whereby you use a mathematical model to predict the probability of an event occurring based on the last event. So for example, a patient comes in and has a seizure, and you can go, okay, well, what's the cost going to be to this patient in terms of the economic cost, but also the cost to their quality of life down the road. And it's a really great idea of, like, so for example, with Google, you go, and you can get any assortment of outputs from that. And the information we put into the model can give us predictions on where that patient could end up and basically the pathway of that patient. Coming on from that, then we're also beginning to do a survey looking at the incidence and prevalence of brain tumor-related epilepsy in Ireland, where we review in adult brain tumor cases the incidence and the treatment of these patients to really, again, get a really good idea of the patient pathway of a patient with brain tumor-related epilepsy. And then further down the line, the hope will be to do an analysis of some other data, which is hopefully going to become available to feed into this, which would be UK-based data. And then finally, we would really like to do a prospective patient survey in Beaumont Hospital and Cork University Hospital, whereby we have a quality of life survey, which we apply to patients who are brain tumor-related epilepsy patients undergoing surgery. So yeah, so a huge thank you to, of course, all of the patients and their families involved, but obviously everybody in all of the teams involved here as well. So thank you.
Questions for Kate? I have one for you.
Oh yeah.
Yeah. I'm just wondering, has there been any effort to develop a drug that targets both epilepsy and tumors? Or is there, that's the first question. And the second question is there any indication that your approach may affect the efficacy of tumors?
Yeah, so without going really in depth into the kind of chemistry of it, there are anti-epileptic drugs which, because of the receptor they bind to, should impact the metabolism of the tumors and impact their, for example, the potential to invade. So the idea being that some of these, like this approach, the idea being it'll block, not only will it block the seizure activity, but it should cause or block an influx of calcium, which should prevent invasiveness and not allow proliferation of the tumor cells as well. Okay. So that's the idea, that it is a dual-pronged approach, that we're engineering the neurons to not be excitable, but that has a feedback approach too, where they then won't produce too much glutamate, won't feed into the growth of the tumor.
And what about, unrelated, what about the interaction or otherwise of temozolomide and any of the other chemo treatments? There's nothing
I don't
that you're worried
know.
about that.
No, I don't think so. I mean, I don't really know yet. No, there's not been a lot of work done in that, in looking at the effect of like radiotherapy, for example, because that you think that might have an effect because of the reactive oxygen species and that will affect it, but there's not a lot of, there's work to be done.
Okay.
Yeah, there's a lot of questions, which I think actually with our in vivo studies will answer quite a few of those, or at least indicate some of the answers to those.
Very cool.
Yeah.
Sorry, Dan, yourself. Yourself, go ahead.
No.
No, you can't ask a question, Heiko.
We do the brain barrier. So you can probably. The other question is, why are you not targeting cells?
Because this is more likely to work, because with this approach, what this is doing is, again, I don't want to go into too much scientific detail, but what this approach does is it creates a glutamate gated chloride channel within the neuron. So yes, we are just treating the symptom, if that makes sense. You're treating the neuron, preventing it from being hyperexcitable, but this method basically works by allowing an influx of chloride when that seizure starts, and that will basically shut down that hyperexcitability because it depolarizes. So basically this approach, I mean, yes, this approach is targeting the neurons and we could target the tumor cells, but in this way, you are shutting down the actual event of the seizure, as opposed to maybe a more long-term preventing them from happening, if that makes sense. Do you know what I mean? And I think if you were to target the tumor cells alone, the potential for them to mutate is quite, it's quite high. They're probably those cells might not proliferate, and then you actually just selected for the population that didn't get transfected. I think that's also part of it. But overall, if we can mop up the glutamate that's present, because the neurons are also producing glutamate, which is triggering the tumor cells and creating an invasiveness too. But if we can stop the neurons from basically firing, they will also secrete less glutamate, and that it's kind of a cyclic process. So yeah, we're choosing to affect one side, but it's also sort of safer to get to just transfect the neurons in a neuron specific way than transfect all the cells and see what happens, because you don't want to stop all your normal signaling either. But this way it should just be when there's a high glutamate expression in just the pyramidal neurons. So yeah, sorry, I didn't want to get so scientific, but that's kind of the yeah.
Sorry. And just from a patient's point of view,
Yeah.
Epilepsy is something that comes up. And I just wondered, because you have some patients who are tried epilepsy, or family members are told that their family member happened to get. And then there are others who've never had a seizure at all.
Yeah.
And I just wondered, I mean, obviously just to say the quality of life thing is certainly something that comes up a lot. And I'm sure we have patients would be very much like to be in that kind of thing, a survey or whatever. But I'm just wondering, do you have a way of predicting?
There is a really interesting trend in basically the development of seizures is inversely correlated to the malignancy of the tumor. So it tends to be patients who have seizures have slower growing tumors. And it's often a lot of the time, well, there's sort of a couple of theories behind it. So if you have a slower growing tumor, you have more time for the tumor to produce glutamate, for example, or produce the factors that are causing the seizures and cause the seizures to happen. Whereas when you have a really rapidly growing tumor, there's not enough time for that to develop. But also IDH mutation is really important. So when you have an IDH mutation, the tumor has slightly different metabolism, and the tumor will produce a molecule called 2-HG. 2-HG chemically looks like glutamate. And so if you have an IDH mutant tumor, which are often lower grade, they actually produce a molecule that's not glutamate, but it looks like glutamate and acts like glutamate sometimes. So actually the presence of the IDH mutation and that molecule that gets produced triggers the seizures also. So that's sort of the trends that are there. So, I mean, there's lots of groups trying to understand really how the IDH mutation works in that way, but the fact that they're just chemically really similar is probably they fit the same receptor, they fit the same molecule. So yeah.
Yeah, actually, you're absolutely right. But tumors associated with epilepsy are absolutely unique and different.
Mhm.
Not all of them are IDH mutant,
Yeah.
and the ones that aren't have large ganglion cells within them.
Yeah.
I know you're recording from the peritumor tissue, but and you got a reading from Kieran speaking there to give you some of the tumor tissue that contains the large ganglion cells. Actually, could record, and that would be really interesting study to
Yeah.
combine the two. You know.
There is another study which PhD project. Yeah, so there will be one of the neurosurgical regs doing a PhD project where that's actually one of the goals of it, to look at tumor, peritumoral and other epileptic tissue to see the differences, because yeah, absolutely, I agree, because it's so heterogeneous, you know. Yeah.
My name is Claire Gardner. I'm a professor in immunology up in Trinity College in Dublin. And I'd like to thank Brain Tumor Ireland for inviting me to present today, because I'm hoping we're going to be working together over a number of years to come, which is great. So I'm an immunologist. So really, the first thing I wanted to ask you was, when you think of immunology, what is the first thing that you think of? And most people, I suspect, in this room probably think of Luke O'Neill, because he's a larger than life character. He's dominated the field of immunology. But the immune system actually is not only important for infection, it's really, really important in cancer prevention and cancer treatment. And this has changed dramatically treatments for patients over the last 20, 30 years. So when we look at now the treatments that are used for patients, which include surgery, radiotherapy, chemotherapy, we now have the molecular precision drugs that we've been talking about in this group here, but also immunotherapy. And immunotherapy is one of the fastest growing drug markets. The pharma companies are mad keen, and they're throwing money at every kind of immunotherapy they can get. I'd love to say it's because they really, really want to help patients, but there's a big market there, because 1 in 2 people will develop cancer in their lifetime. So they have a very big incentive in investing in it. As an immunologist, I'm delighted because the immune system has not been exploited as it can be for therapy. And the immune system, all these immunotherapies, were only at the beginning of what is possible. So I actually think the future for this is really, really exciting. And we know that immunotherapy is already the standard of care for many different kinds of cancer types. So there's a huge number. When people talk about immunotherapy is just massive. And the types of drugs and the field of immunotherapy is absolutely huge. And that's mainly because the immune system is made up of so many different molecules and so many different cells and so many different parts. And every single one of them, immunologists are thinking, well, how can I use that to fight cancer? Or could I use that to fight cancer? So what I'm talking about today are kind of cell-based immunotherapies. So by that, I mean, we're talking about immune cells. So your white blood cells, so in your blood, you've got red blood cells, carry your iron and oxygen. Your white blood cells are your immune system. So even within your blood, you've got lots of different types of immune cells. So what I'm talking about is just one of these or two of these. And these are called either T cells or NK cells, natural killer cells. And the reason these cells are so important in cancer is because, one, they protect us when cancer arises in us spontaneously, because cancer does arise in people all the time, but we don't necessarily go on to develop a severe cancer or a tumor or whatever. So the normal job of the immune system is to recognize tumor cells that arise in our bodies and to kill them. So if they meet a cancer cell, they're in our bodies circulating around. If it meets a cancer cell, it can kill it. And that's why, in addition to their normal purpose that they naturally do, we can target and use these cells in patients' bodies, or use cells from a third party, but use these immune cells to kill cancer cells. That's what they're designed to do. That's what they evolved to do. And that's what lots of the cell therapies are now targeting. So what happens when to the immune system as cancer progresses? So you'll have to bear with me, I'm not very artistic, and that was my car. That was the best I could manage on PowerPoint. But in my head, I was trying to explain it. So if you think of the immune system as a car, in a normal, healthy individual, you've got a good immune system, you've got a good working car. And that car is very fuel efficient. It's past its NCT. It can drive on the motorway at whatever the speed limit is. It's comfortable. It can get you from A to B in a minimum of time. So that's a healthy immune system. So that's our healthy person. What happens, as I said, cancer cells do arise in people. And I can't remember, there was some mathematical working out, but it was something like, I don't know, 10 cells per day or whatever it is, can spontaneously arise in people. But we don't get cancer because the immune system can get rid of them. So if you encounter just one cell, the immune system, and you have a good working immune system, it's like a little speed bump. It'll just slow down briefly, deal with it, and get on with it. Doesn't slow down, keeps going. Okay, so it's able to keep going. If you get a group of cancer cells that start to develop, then they start to attack the immune system. Then you start to get problems for your car. So if you imagine the equivalent of somebody coming along and throwing a little bit of dirt into your oil, into your engine oil, you're just slowing down the car. It can still drive, but it's not working as well as it should be. Somebody comes along, chucks a stone through your window. Again, it's not going to help you drive. So you can get an accumulation of these different things that can happen, which actually can impair your immune system. What can happen then is, as the cancer cells grow into a tumor, they increase the number of ways in which they can damage the immune system. So steal the fuel, break your windows, we said, slash your tires, whatever you want. And you get to the point where the immune system is paralyzed and it can't go. So your car can no longer move, your immune system is broken. So the goal of immunotherapy, or the approach that we take, is we want to understand what is going on with the immune system. We want to understand why it's broken in cancer. And then when we understand specifically how it's broken, we can then look at ways in which we can actually fix it. So you're probably familiar with pembrolizumab, you may have heard of. It's one of those blockbuster checkpoint inhibitors. So what I would say, that is like in our broken car, if your tire is slashed by the immune system, you come along and you put on a new tire and it can go again. It's not perfect, but it's moving better than it was. So those different kind of immunotherapies can target different bits. Now, one of the big issues is that not all cancers are the same. And the damage that different cancers do to the immune system is different depending on the cancer that you have. So, for example, in one cancer, they may target breaking the windows of the car. In another one, a different cancer type will slash the tires. So you can't go in with necessarily one approach for immunotherapy for all different cancer types. You have to figure out what is specific for a given cancer type and then develop something like that. So what we're going to be working on, and I'll tell you about this new project that we're doing, is specifically focused on glioblastoma. And so what we want to understand is, in patients with glioblastoma, when you have this tumor, what is going on? What is this tumor doing to break the immune system? And in turn, how can we use that knowledge to then fix that immune cell? So the project, again, I would agree with Annette, getting funding is the hardest thing. And we may have many great ideas, but until we get funding, we're unable to do much. But anyway, the good news is we're post-funding, which is brilliant. But we have put in a grant to the EU. So we got funded to the tune of 1.2 million. So this is your European tax dollars at work, so go EU. And it's to work specifically on glioblastoma. So again, what we tried to do, and just one really nice thing is that we're leading the research as well. So it's not always, other countries being experts. We're the ones who are leading this project on glioblastoma. So there are 4 countries involved: Ireland, Germany, Norway, and Belgium. And again, myself and my colleague David Finley, we had tried to identify people who had expertise in things that we didn't have that were complementary to our stuff, so that we'd be able to maximize the information that we can get on these tumours. So these 4 countries are involved. The Irish portion of the funding is about 370,000. So we're able to take on 1.5 people to do the work. So great. I know. We have to interview both. See, but in Trinity we have myself, David Finlay, and 2 new people on this project. In Beaumont Hospital, the team have been really, really helpful over there. So a colleague of Kieran, so Steve McNally is helping us with this, and Patrick Morris, and Eloise Cowie, who's the nurse specialist out there. And also Keith Egan is helping us with the ethics and the paperwork and everything that we're going through. Of course, the patients are going to be coming from Beaumont Hospital. So that is a very, very important link, because we really, this work is all about patient focused research. We're also hoping that Brain Tumour Ireland will be part of this study, in part because we need patients and healthy controls. And in our work, looking at patients is not enough. We have to be able to compare patients to healthy individuals to see what is wrong in the patients. Okay, so we have to look at healthies to understand what's normal, and then look at why the patients are different. So we're starting a campaign where we're currently applying for ethics in Trinity College, but to try and email people who are associated with the charity and ask for volunteers, for healthy donors who would be willing to give a blood sample for this research. So hopefully we will be back in contact with some people soon. But it does give a chance for families, members who sometimes can feel quite useless in terms of making an active contribution, that it is something that people might be interested in doing. So in terms of specifically, what are we going to do and how are we going to do this work? We're going to analyse blood and tumour samples from patients with glioblastoma. So patients will be recruited when they come to Beaumont Hospital and they're undergoing surgery. And a sample of the tumour will come to our lab, and then what we'll do is a combination of, we'll look at some of the immune cells that are present in that tumour, and we'll characterise them. Again, we're particularly interested in the cells that are good at killing. So these NK cells and these T cells. And very similar, I suspect, we're going to collaborate on that we know as well with glioblastoma that they release a wide range of small molecules and metabolites. We're particularly interested in some of those and how they have a negative impact on the immune system. So for example, we can find that, you can get all of these different molecules that are present, but only this yellow triangle is the one that's going to turn off the immune cell that we need to do a particular job. So the first thing is to understand the factors that are being produced in the tumour. The second thing is to understand how they impact on the immune system. And then the next thing we can do is we have the technologies. One of the biggest advances which has facilitated immunotherapy has been nothing to do with the immune system. It's been to do with gene editing, how you can change a cell, how you can put a new molecule into a cell. What we can do, we can take out molecules. So basically, if we know that they have this little yellow thing on them, we can basically interfere or zap the cells to take that off. And then we can restore function to those immune cells, which can then be used for patients. So that's the first thing. The second thing is, and this has come up, was briefly mentioned by somebody earlier, so we're at the moment, the first part is to look at the immune cells that are present in the tumour. Now, if we want to restore function and use them as a therapy, that's not going to be a viable option, because by the time you get a tumour out of a patient and isolate all the immune cells that are there, you're not going to have enough cells for a viable therapy. But what we find is that if you look in the blood of the patients, so the same patient with the tumour, and we look in the blood, and we will be doing this as part of this study, we find that it's almost like a gradient of defect. So you've got the cells that are the immune cells in the tumour are completely paralysed. The ones in the blood are 75% paralysed. Okay, so you can get work with these cells that are present in the blood, and we can get a lot of those immune cells from the blood. We can harvest a lot more. We can work on them in the lab and give them back to the patient. And our idea would be, we would be giving them IV. So I know people will talk about methods of delivering, and there are ways of getting cells into the brain directly and so on. But again, there is evidence that that leaky blood-brain barrier can be present during glioblastoma as well. And in fact, there have been studies recently which have shown that immune cells, which are normally only ever found in the circulation, are found in the brains of patients with glioblastoma, including these cells. So we're hoping that we will be able to get them. I kind of think that's somebody else's problem if it doesn't work fully, but there are ways to do it. So that's kind of the goal, is to look at a combination of tumor tissue, immune cells in tumor tissue, to look at the blood, to look at the immune cells in the blood, understanding how to fix them, making a product. And some of our collaborators within the EU, for example, the Norwegian group, they make these cell therapies in a proper GMP facility or whatever. So they have the technology and the scale up feasibility for some of this work. One of the reasons that the healthy controls are really important is that when we look at patient blood and we're trying to figure out those in the patient's blood, why are those cells not working properly? We can look at them and look at the different molecules. And in this case here, we can say, well, they've got these red bits and they've got these yellow bits. So which bit should we target? Well, the point is you don't know until you compare it to a healthy. So if you look at the same cells from a healthy individual who doesn't have cancer, you can see that they all have the red. So the red is actually normal. So that would be a really bad target to go after. So we have to look at, compare the healthies to the patients, and then that will help us to pick which is the right target to go after. So I just briefly wanted to mention CAR therapy because I know it's very topical at the moment, and a lot of people have questions about it and what it is and how it works and can it be done for glioblastoma. So in my head, the simplest way I have of thinking about this is if we go back to our car and we have our immune system, if we have a sat nav in the car, it's like giving the sat nav an air code. Okay. So in Ireland, before we had air codes, you used to say, when you turn left and you turn right, and then you pat the pole, there's, take 3 lefts and you'll be grand. Now you give them an air code and it'll bring you exactly to one unique location in Ireland. And that's what CAR therapy does. If you can find an air code that is unique to your cancer, then you can send it. And some cancers have that air code. So like lymphoma has that air code, HER2 positive breast cancer has that air code. So any of those cancers, there are some that are quite easy and suitable for this. For GBM, so far, there is no unique air code available. So what we have are kind of what I would say partial addresses. So we know, for example, that glioblastoma cells are found in a place that has KIL in it. Okay, unfortunately, it's a very common thing, the word for whatever, a wood, is it in Irish? Okay, so you have Kilkenny, Kildare, Kilmuckridge or whatever. So with that partial information, you can send the immune system to here, here, and here. It narrows down the country, but it's still going to 3 different places. So it's not a specific one. And this is currently where glioblastoma is in terms of CAR therapy. But again, I want to tell you that even with this not perfect air code, these are just some of the trials that are going on at the moment. And this website, clinicaltrials.gov, it's fantastic. You can just put in CAR cell glioblastoma and it'll tell you every single trial that's going on. But the point I wanted to make on this one, in red, I've highlighted these are the air codes that they're using, and they're all using different ones. So there's no specific CAR that we can say right now is going to be really good for glioblastoma. But we have several partial addresses, and the way things are going, actually, I think cell therapy is a viable option and will be tested in patients. So I'm going to stop there and take any questions, and apologies if it was too simple. And I'd like to thank the EU for their funding for the work.
Any questions for Claire? Yeah.
Yeah. I was just wondering, what would be the best approach to really try and identify these tumor-specific antigens, especially for like such a heterogeneous tumor?
Well, you see, I think that's one of the challenges
Yeah.
with glioblastoma is that it is very heterogeneous. So, and I think that a lot of people have tried to identify, and all what they've found is things that are enriched. So nothing that is specific. And as you know, and I know, that part of the problem with that is that if it's not specific, that means it's on other cells. And so you're going to get immediately a large number of off-target effects. So people have tried and haven't found a specific marker. I don't think they're going to at this point. So it may be a combination of things that they would use. So for example, you know, rather than go all out and target one of those markers, what you might do is use a suboptimal version of that, so that it's only when you get the combination of three things recognizing that you turn on the full activity of that cell. So, yeah.
One of the ways to overcome that might be to include a control group of non-GBM tumors that are lower grade, that will not have the same breakdown of the blood-brain barrier, might in fact have more of an lymphocytic infiltrate. So a control group like that might have to answer that question.
It might indeed. Yeah. I would agree.
So, one thing I guess it's more of a comment and an offer would be, I mean, if you can recall Kieran's presentation, we've identified different TME, just TME environments, with one of them being specifically immune hot, if you like. And we would love to do RNA sequencing analysis of your tissue and be able to inform you which of the immune hot, cold, and medium to relate whatever you find in terms of your signaling pathways to those subtypes.
Yeah.
Because you may find ultimately when you go into the clinic that the immune hot cohort are going to work better in whatever treatment you make.
Yeah.
So I'd look to stay for that.
Yeah, no, absolutely.
The other question, I think, is a broader question, is how do you get over the fact the big problem with GBM is it's an immune cold, and it's just escaping everything. How are you guys going to get over that?
So there are a couple of things. One, NK cells have been found in glioblastoma tumors. So it's not like they're not trying to get there. They are trying to get there. So again, one of the things that we can do with these cells is engineer them. So again, we're not going to be able to give them a specific tumor marker to go to. But for example, we know that in the tumor environment, and you got the update on this, they will produce certain chemicals like a chemokine or whatever. So what we can do is we can engineer the immune cells with a little signal in the lab. So when we put them into patients, they will respond to the signals that are being produced by the brain and will naturally traffic up there. So we have ways in which we can engineer them to get there. Okay. And then again, one of the things that we're really looking at is when they get there, how do we prolong their function there? Because as soon as they get there, they're beginning to, the environment isn't very suitable. You've immunosuppressive things there. So trying to understand those and basically pre-arming the cell so that when it gets there, they won't turn it off. So yeah. And some of, like, again, the groups that we're collaborating with, there's a molecule called TGF-beta, which we're familiar with, but this is a very immunosuppressive chemical that can be produced in the body. And she has a novel mechanism of an antibody therapy that's actually in trials in patients in Belgium but it's a way of inhibiting this thing without inhibiting the entire immune system. So we have lots of ideas. What we just want to do now is try and implement them. And the group in Germany, they have a really cool thing where they have a really big tumor bank over there, and with lots of glioblastoma tumors, and they can tissue section them. So take a tumor and just put tiny little slices on different slides and put a slice on each slide. And then they do this fancy analysis and they can tell you every chemical that's in there and where it is. And then they layer on the second one and they can build up a 3D model of a tumor and then tell you where exactly every chemical is in there. So like they're running out of glucose there, or once they hit that region there, that's when they stop working, and so on. So again, when we're working together with them, I'm really hoping that we'll be able to build up a real picture of expertise. That's very exciting.
Thank you very much, Kerry.
I'll be giving a talk on why we're discussing about drug delivery and techniques. Naturally, what's come up before is a lot of issues regarding the blood-brain barrier. So we start off with the optimum conditions for treating a brain tumor, which would be to achieve sufficient quantities or concentrations of an efficacious agent within the tumor, but with minimal systemic side effects. And that would be the optimum type of treatment. However, we're faced with two problems. One is that we've constantly mentioned the blood-brain barrier. The blood-brain barrier, which I'll get into a little bit detail, is a very highly selective semi-permeable barrier. The whole point of the blood-brain barrier is to create a very privileged environment, both from a electrochemical, metabolic, and immune to environment for the neurons to allow them to fire their synaptic firing. And that's the whole point of the systemic delivery of simply oxygen, glucose, and a few nutrients to these neurons to allow them to function. And the second problem with delivering tumors into any, or delivering drugs into any form of tumor, is that there's high intratumoral and peritumoral interstitial fluid pressures. So there's two main problems that we're trying to face when trying to get adequate concentrations into a tumor. The blood-brain barrier, the brain is a phenomenal organ. It receives about almost a fifth of the cardiac output for an organ that weighs about 1.2 kilos. And the picture on the left is actually a latex-injected vascular model of the brain with the brain tissue removed. So that's the vascularization of the human brain. And behind that vascularization is a protected and very unique environment for neuronal activity. There's two aspects to the brain's privilege. It's the blood-brain barrier, and then how that barrier is coupled to neuronal activity. And so the blood-brain barrier is formed of three units. One is the endothelial cells, and they're ubiquitous throughout the body. And these cells form tight junctions, and that pretty much forms the lining of the blood vessel. There's the periocyte that wraps itself around it, and it's a specialized cell that actually talks to the endothelial cell on how to form these tight junctions, but also regulates immune entry in and out of the brain. And then there's the astrocyte. The astrocyte is the go-between cell. It's a glial cell, and it's the go-between cell between the neuron and the blood-brain barrier. Then the neuron is coupled to this unit through the neurovascular coupling. So, for example, the neuron can call forth any form of blood supply it requires. For example, if you were to give me a task to look at a sum and to calculate, there'd be certain parts of my brain that will be recruited through a neural network. And these areas will require increased glucose consumption to run this program. And the way the brain modifies and brings forth the glucose and oxygen to do this task is through the coupling mechanism of the blood-brain barrier to the neuron. As I mentioned before, it's a very privileged environment, and so pretty much the only thing that gets into the brain is oxygen, glucose, a few building blocks, and very, very small lipophilic molecules that can just transfuse or diffuse down concentration gradients. And that's pretty much the only thing that gets in. From an immunological point of view, the parasites control the systemic immune system, and the brain has its own microglia and that acts as its immune guardian. Now, the term glioblastoma disguises the fact that it's a tumor of an astrocyte. And the astrocyte is part of the blood-brain barrier. So whenever we talk about gliomas, there's two types of gliomas, either the astrocytes or the oligodendrogliomas, but the predominant, when we talk about glioblastoma, it's all astrocytes. So it's a tumor of the blood-brain barrier and one of the core components of it. And whenever us the surgeons look at a tumor like this, and the tumor can be divided into 3 zones. This is a dense necrotic zone. So there's a metabolic and cellular gradients from a dense necrotic anaerobic environment to a permeable neovascularized angiogenic to a peritumoral region of the brain. Here, there's no functional tissue. So as a surgeon, we can just remove all this and be very disappointed with anything less than the 90% resection. So this permeable part of the tumor we remove at the time of surgery. What's left behind is the non-enhancing tumor that's behind the protected blood-brain barrier. And therefore using systemic therapies to deal with what we leave behind, because we remove the easy bit, what we have left behind is the more difficult bit. So what we have is the non-enhancing, fast-growing tumor. It just has a different phenotype. It hasn't had a chance to set up a dense necrotic center yet, behind a protected blood-brain barrier. So we're faced with 2 problems. We have a tumor bulk. This is another tumor, and you can appreciate the shift. So we can see the mass effect. This is increased cellular growth is causing mass effect. This is a physical phenomenon. The brain is a viscoelastic substance, and fluids will flow from pressure from areas of high pressure to areas of low pressure. So what we see is a shift from an area of high pressure to areas of low pressure. And what happens there is this is a combination of cellular bulk, but not only that, the angiogenesis, increased blood flow, and the leakiness of the intact tumor that rises in interstitial fluid pressure. So what we're trying to do is get drugs into this mass, down concentration gradients, but against interstitial pressure gradients. So what the current drug delivery systems that we have is oral temozolomide. And what that is, it's a tablet that's ingested, it absorbed through the stomach, goes through first pass metabolism, and then into general systemic circulation. So what happens is every cell in the body, every organ receives the same concentration extracranially as every other cell, except for the brain. Temozolomide only gets into the brain because it's of a low molecular weight and it's lipophilic. So it will diffuse down. But what you're trying to do is diffuse it down a concentration gradient and up an interstitial pressure gradient. So large amounts of systemic temozolomide need to be given to create this favorable environment for this very physical molecule to get into the tumor. And so what we have is, because of we can either hijack the blood-brain barrier's uptake mechanisms, and there are several things like endocytosis and pinocytosis, or we rely on the physical property of the molecule to get into the tumor. So people have been for a long time trying to figure out how can we achieve the optimum goal, that is high intratumoral concentrations with minimal systemic effects, and everybody keeps coming back into a local delivery system. And throughout the world and for many, many years, people have been trying different things. So this is just an infographic from a recent review article where there's different types. And the most common type is the stereotactic placement of either a needle or a catheter system. So the stereotactic injection will deliver a single one-off shot into a location in the tumor. The one below it here is the convection enhanced delivery. So that's an indwelling catheter. And what that is doing is creating, I'll go into each one actually individually. So the first one here is focused ultrasound. So what this strategy to get drugs into the blood-brain barrier is about blood-brain barrier disruption. So using focus and energy in the form of sound waves, and everybody understands what an ultrasound is. It's just simply sound waves. If you create a certain sonification, at certain frequencies and timings, you can cause disruption in the blood-brain barrier, and you can turn an impermeable barrier to a more permeable barrier. And there's theories that it can work both ways. So there may be roles for liquid biopsies in tumors later on using this technique, and also getting drugs in exploiting this temporary disruption to the blood-brain barrier. The next one is convection enhanced delivery. And again, this is another cheat around one of the problems, and that is the interstitial pressures. So the convection enhanced delivery is an indwelling catheter into a solid tumor, and it's connected to a syringe driving pump. So you create a pressure gradient in an attempt to overcome the second problem of drug delivery into this. And this has been employed in diffuse pontine gliomas, currently known as diffuse midline gliomas, which is an incurable and inoperable tumor. But again, both the convection enhanced delivery and the focused ultrasound are just cheats at trying to bypass the problem. A novel method also that's being recently used is intranasal delivery. And that uses the fact that in our nasal mucosa, we have nerve endings that are bare and naked. And we have, through the olfactory and trigeminal pathways, there's a window into the olfaction areas of the brain. So these drugs delivered this way can get into areas around the olfactory cortex at the base of the brain and quite close to motor areas, extrapyramidal areas. And the theory is that from that point, then there may be a wider distribution through the lymphatic system, which is a newly described system. But the solution from our perspective is, surgical resection. So at the time of surgery, when you remove the core, T1 enhancing, that bright white area, the permeable area, you're left with a cavity. And it's at that time you have that cavity, surrounding that cavity, you have protected residual tumor. So it's at that time that there's the first opportunity to deliver many things directly into that cavity. And also surgery will drop the interstitial pressures in the surrounding tissues, but you're still faced with an intact blood-brain barrier. And that's where the local delivery should come into place. So the bottom graphics are actually very interesting. In yellow, you can see tinzolamide and the way it peaks, and that's to do with the tinzolamide cycles. Blue is the old carmustine wafers. It's a BCUN alkylating agent, which is similar to tinzolamide, a little larger. But these wafers, which were very large tablets, were placed into the cavity of tumors, and it would give a one-off dose of local therapy. But then, due to poor application, there's a few side effects. But the ideal one of local delivery is where you have a consistent and sustained concentration, and which should translate into efficacy with very little systemic toxicity. And to achieve sustained concentrations over time, you need to design what you're bringing in there and how you deliver it and how it's released. So time-dependent release of certain agents can be achieved through the formulation of nanoparticles. And nanoparticles need to be carried in naturally biodegradable gels, which we inject. So a nanoparticle is essentially a very small envelope, and that can package anything. And you can exploit either the blood-brain barrier's own method of transporting things from the outside inside, or it can be tagged and targeted and for an active uptake mechanism. And that's that.
Questions for Kieran? I have a question. How do you determine when you're operating, how do you determine how far you go with the resection in any given patient?
So it depends on the functional location of where the tumor is. The sad thing about gliomas, they can be very easy to operate because essentially you've got a dense necrotic non-functional core. The white outer area is angiogenic. There's going to be very, very little functioning tissue in there. It's the tumor that's left behind in the infiltrated brain behind, that's infiltrated functioning working brain. So because these are such a rapid destructive tumor, there will or will not be a functional deficit due to that necrotic destructive tumor. And as surgeons, if you don't focus on the tumor and its biology, all you do is clear out the necrotic area, and all you're doing is clearing out dead cells. You change the metabolic environment very briefly, but if you leave any T1 white bits behind, or naturally the vast majority of the time we're leaving non-enhancing tumor behind the protective blood-brain barrier. So to answer your question brief, it depends on the functional location. So whenever I plan surgery, I do try to go supramarginal, which is outside the T1 enhancement, and I will define zones that are functionally possible and not possible.
So you determine that before you operate?
Yeah. But the most important thing is not to create a functional problem, because then you're limited with quality of life.
Yeah.
And then if it's substantial, then full treatment may not even be possible.
I would say, I would agree with that. At that time of surgery, would that be a good time, for example, to put in a cell immunotherapy?
Yeah.
Because I know there are a lot of people, like, part of the problem with the CAR T cell stuff is that it has to be personalized for a patient. Whereas there are lots of other therapies, cell therapies, some NK cell drugs that are more being designed as an off-the-shelf therapy.
Yeah.
So that you could, for example, be able to, at the time where you surgically resect, to actually put activated immune cells in there as your first line of therapy.
So, I mean, the answer is yes. I mean, it's quite conceivable, and we're hoping to develop a catheter-based system where an indwelling catheter you can load with standard of new care, something novel, but ubiquitous, and to try and target as much. Once you've analyzed the tumor, you can then develop a more personalized and come back and then tailor through repeated injections. I mean, that's a very possible situation where you start off with a broad spectrum of treatments, and then you start to narrow down once you've done the full in-depth analysis, which can take many weeks, looking at mutational burdens, etc., and weaknesses, and then you come back with a more personalized approach.
Sure, that's great talk. This is probably the last 40 years. After you resect the tumor, as you're quite right to say, you drop the pressure inside.
Yeah.
That, there's now a hole, and for anything to go from that hole at normal pressure out, say 2 centimeters beyond the margin there, is the technical problem. I can't see how it can be overcome. How can you drive something 2 centimeters beyond that hole into the surrounding brain where you know the tumor is going to recur? It's all going to recur in the same place.
So
that's the big problem.
So there is a limitation. I mean, naturally, there's all limitations to every strategy that we try. The diffusion distances of different molecules, depending on the strategy that you're going to use. So if you rely on, for example, a gel-based system with free tinzolamide, you're going to rely on a concentration gradient and the physical properties of that molecule to get as far beyond the interface and deep into the remaining brain. That at the same time, you could have tagged nanoparticles using active uptake mechanisms. And if you can find a way to exploit the lymphatic system as well. So where there's a CSF around the periarterial spaces, you get in there, you start to spread out further. And that's why the intranasal delivery systems have started to shine light on the lymphatic system and how you can get access to one region in the brain. And from there, once you get access a road, a map, a weakness, or it can spread out through different areas. So
sure in most models of glioma, is it?
No. Well, I mean, there's yes, not in mouse models of glioma, but in other neurodegenerative disease processes. So that lymphatic system has been described for motor neuron disease, movement disorders, but for glioma, I mean, why can't we exploit it for glioma?
Can I ask a question on that as well? Sorry, I'm thinking again about cells. So cells are quite different from drugs in that cells can physically move themselves. They don't have to go along with a concentration gradient in the same way. How does interstitial pressure affect cell migration? Do you know? So
it's been a long time since I've looked at this, but I mean, my understanding is, for example, if you were to take the blood vessel and the endothelium and the laminar flow, along the cell adhesion, there's molecules that will react to blood flow and create an intercellular pathway and signaling. So by the same method, the shearing forces inside a large tumor can create intracellular and extracellular changes. And so that doesn't really answer your question, or no? No.
I just know that, like, I'm thinking of, say, the gut or whatever, like you can have immune cells that are, you know, in this, or blood vessels, and the blood is flowing really fast, or they're complicated, but the immune cells can still slow down, stick to a vessel wall, migrate. You know, they've evolved to do that. I'm wondering, might they be able to overcome some of the?
Well, not really. I think we've seen lots and lots of high-grade gliomas over the years where the lymphocytic infiltrate. It's always at the periphery of the tumor. You don't actually get into the tumor. They're always at the interface between the brain and the edge of the tumor. That's really sad. And we always thought 100 years ago that those that had lymphocyte predominant phenotype always did a bit better anyway. You know, they never get into the tumor. They're always on the edge, surrounding it. So.
Hey, thanks very much. Thanks, Kieran. That's great.
Thank you. So good evening. My name is Fiona Keegan. I'm Chief Executive with Brain Tumour Ireland. It is my great pleasure to introduce the inaugural Ronnie Feely Medal Award. Ronnie, whose photo you can see there, in 2011, at the age of 62, was diagnosed with a grade 4 glioblastoma and sadly died 3 months later. Suffice to say, Ronnie's family, many of whom are here this evening, found it really difficult to access the support they needed and the practical information they needed following Ronnie's diagnosis and throughout her treatment. And following Ronnie's passing, the family came together and decided that they were going to try and fill that gap. In 2012, the Ronnie Feely Foundation, also known as Brain Tumour Ireland, was established. Brain Tumour Ireland has established this award not only to honour the memory of Ronnie herself, but to honour the tremendous work and dedication over the last almost 11 years shown by Ronnie's family. And that has brought our charity to where it is today. Boards of directors will come and go, CEOs will come and go, but this award will ensure that the reason this charity was started will never be forgotten. So, to the award itself, we invited those at the earlier stages of their work in research to submit a 400-word abstract giving an overview of a brain tumour research that they were working on either currently or have worked on in the past, and to design this abstract in such a way as to be understood by a lay audience. I understand that Ronnie was a pharmacist, and her daughter Natasha told me that she spent most of her time on the other side of the counter, explaining the medications and trying to ensure that people understood what they were taking and why they were taking them. So we like to think that Ronnie herself would approve of this award in her name. We had a large number of submissions from right around the country, and all of an extremely high standard. But as they say, there can only be one winner. Not one for everyone in the audience, just the one winner. And that winner is Mr. Florian Oswald. He is a final year student at the University of Stuttgart who's completing his master's research in the Department of Physiology here at the Royal College of Surgeons. And I now invite Florian to give a 10-minute overview of his winning submission. And then I'm going to invite Ronnie's daughter, Natasha, to award Florian with the Ronnie Feely Medal for 2023.
Thank you for the introduction. And yeah, today it's my great honor to present you a project I was working on the last few months, and it's called Investigating Treatment-Resistant Mechanisms in Glioblastoma. Also prepared a little introduction slide. There you can see me hiking in the lovely Irish rain. And yeah, so just got introduced. Yeah, I did my bachelor's in biotechnology at the University of Stuttgart, and now in the master's program. And for the last and final part, I got a chance to work here in the group of Professor Brona Murphy on glioblastoma. So glioblastoma, or short GBM, is a brain tumor, and every year in Ireland, around 300 people get diagnosed with glioblastoma, and in comparison to the United States, it's around 17,000. How we treat glioblastoma nowadays, so the standard of care. I try to illustrate here the tumor with the different cancer cells. And there are two major treatments. So we have radiation therapy and chemotherapy. And the most common chemotherapeutic is called temozolomide, or short TMZ. And both of these treatments, they try to kill the cancer cells. So if we treat here, we can see that these cells, they died, but some of them, so here I'll try to illustrate in the blue, they survived the treatment. And even if we treat with more temozolomide, they still survive, and after a few months, they will grow back, and we have what we call a recurrent tumor. And this recurrent tumor then consists of cells which are resistant against temozolomide. So if we try to kill them now with our chemotherapeutic, they have kind of this protective shield, what I want to show, this red circle here, and we have no effect anymore. In our laboratory, we work with glioblastoma cell lines. They are from patient-derived, so after the removal of the tumor, we get the cells in a little tube, and then we can culture them in the laboratory and perform then different experiments with them. In a previous PhD student of our group, she kind of simulated a clinical treatment. So she cultured cells in the laboratory and treated them over a few months with temozolomide. There she could observe that some of the cells died and some of them, they recurred. And we had like this recurrent tumor in our laboratory. And what was the aim now of this project, of my thesis, is to study the changes in the temozolomide-resistant glioma cells to better understand the mechanisms of treatment resistance. So just to make it, so we want to see what is the difference between this original glioblastoma cells and the resistant glioblastoma cells. And therefore, we use different methods. So one special thing in our lab is that we culture them in 3D. So we'll talk about that in a minute. Then we measure the effect of different drug treatments and analyze the genetic information as well as the proteins. So as I just said, we culture them in 3D. So we have kind of glioma spheres. So that means like normally in a 2D cell culture, the cells, they all grow on one surface. But here they attach to each other and form kind of a 3D construct, which is kind of looks like a tumor inside the brain. And so that's like many advantages. So we can like, we can like replicate how the cells, for example, communicate with each other. When we treat the cells, kind of the cells on the outside, they get a higher concentration of drugs in comparison to the cells on the inside. Then we treated our cells, so both cell types, so the original glioblastoma cells and our resistant glioblastoma cells with different drugs. So therefore we first have here an untreated control. So here the orange bar is the original cells and the blue, the resistant cells. So we just let them grow over a few months in our a few days in our lab. And in another sample, we had the same cells, but this time we treated them with temozolomide. And then after the 3 days, we could observe that when we treat them with temozolomide, only 70% are still alive. So we kind of killed 30% of the cells. And in the resistant cell lines, there are, as we expected, there's nearly no change. Then we treated the cells with 2 other drugs, which are also investigated in our lab. So if here candidate drug 1 and 2. And we can see that here, if we look at the original cells, and this orange bar, you can see that we have a way better effect in comparison to temozolomide. So for both candidate drugs. But when you look at the resistant glioblastoma cells, so here in the blue bars, we compare now just for candidate drug number 1, we can see that the effect is not that good in comparison to the original cells. So we think, or we hypothesized, that we have this temozolomide, or this achieved temozolomide resistant cells, but this protective shield is not only against temozolomide, it's against like all different types of drugs. So we have kind of a general treatment resistance. And we want to investigate it further and analyze the genetic information. And therefore, we took both our cells, so the original and the resistant cells. Then we kind of broke the cells up and isolated the genetic information. And then we analyzed it, and we could observe that there's a high activity of genes, so or this high activity of survival genes. So it means that like the parts of the DNA which have the information for survival, they are like highly active. And then we also observed the proteins. So we kind of did the same. So we broke up the cells and we isolated the proteins inside the cells. And there we observed the same, that we have a high amount of proteins in the resistant cells in comparison to original cells, which are pro-survival. So this kind of counteracts the killing signals from our treatments. So yeah, how does this work? So we have here our resistant glioblastoma cells, and here our pro-survival protein. And then we try to kill the cells with our drugs. So we get, the cells get like signals to die, but this protein binds to this signal pathways and thereby prevents the cell from dying. But it was shown that in other types of cancer, the same protein is upregulated. And there are drugs in clinical use. And the idea behind it is that this drug, which here is the drop, it binds to the protein and thereby prevents it from working. And then when we try to treat the cell with our different drugs, which induce like the killing pathways of the cell, they can work and the cell will potentially die. So yeah, that in conclusion, so we find out that the temozolomide resistant glioblastoma cells, they're also more resistant to other treatment. So they have a general treatment resistance. We observed that there's a high pro-survival activity, so on genetic information and also on protein level. And yeah, in future work, we have, of course, so we kind of simulated all in the laboratory. So we have to analyze it in patient samples that we could observe the same. And then, yeah, we potentially can examine like the best combination therapy. So do we have to treat like both at the same time, or probably pre-treat with one drug and then treat with another drug? So yeah, thank you especially to Brain Tumor Island. Such an honor to stand here and yeah, present my work and for this award. And yeah, thanks to all the patients and their families. Thanks, of course, to Professor Bruna Murphy, so give me the chance to work in her group. Yeah, it was really good to work on this project. And of course, yeah, to my two lab mates, Sean and Neve, also here. It's also, yeah, every day was a pleasure to come into work. And yeah, thank you also for SFI for funding this project. Yeah.